In this topic, we are going to understand how the bacterial cells are made competent to take DNA, or in other terms, what are the various methods by which the DNA fragments which are isolated now incorporated into host cells. So here we are going to study the various methods by which the DNA is incorporated into the host or the bacterial cell. Now DNA is a hydrophilic molecule. It is hydrophilic because it has sugar phosphate backbone, which makes it hydrophilic, negatively charged. And since it is hydrophilic, it cannot pass through the cell membrane. There have to be certain methods by which it can be then made to enter inside the bacterial cell wall so as to be a part of that recombinant DNA. Now, in order to make bacteria to take up the plasmid, the bacterial cells must be made competent to take up the DNA. Now, there are various methods. The first method is we take the bacteria and it is treated. So, it is treated with the concentration, with the specific concentration of a divalent cation, that is calcium chloride. So, first method is by treating bacteria with specific concentration of calcium chloride. Now, when we treat with the calcium chloride, that is the specific concentration, it increases the permeability of the bacterial cell wall. So, what is it doing actually? It increases permeability of bacterial cell wall. Now, after the bacterial cell wall has increased permeability, it is then subjected to heat shock. That is, this bacteria is then subjected to heat shock. And this heat shock is given by putting the bacteria for a very short span of time at about 42 degrees Celsius. So at 42 degrees Celsius, these are given heat shock, and again putting them on ice. Putting them on ice. This is the cooling method. Now this enables the bacteria to take up the recombinant DNA. Now recombinant DNA is taken up by bacteria. So this is the first method by which we are able to incorporate DNA into the bacteria. Now, the second method is the use of micro injection. So the second method which we are going to use over here is micro injection method. Now, in this micro injection method, it is basically a technique in which the rDNA is directly injected into the nucleus of an animal. So over here, the DNA, that is rDNA, recombinant DNA, is directly inserted into the nucleus of an animal cell with the help of micropipettes or micro needles. So here suppose if this is the nucleus This is the nucleus. The micropipette. This is the micropipette. And it has rDNA, which is directly injected into the nucleus, and this is now ready. That bacterial cell is now ready to take up the DNA. Now the third step, the third method which we use is ballistic or we call it as a gene gun. This is the third method which is being used to incorporate DNA into the host cell. Now in this method, it is mostly suitable in case of plants. Now in this over here, the cells are actually bombarded with high velocity micro projectile of gold or tungsten, which are coated with rDNA. So what is over there? We are using microparticles of gold or tungsten. These are inert, almost non-reactive. So they are carrying that rDNA. That means these microparticles are carrying or are coated with the rDNA and are then bombarded with high velocity into the DNA. That is into the bacterial cell, and thereby the bacterial cell takes up the rDNA. The fourth method is the transfection. Transfection, it is the fourth method. Now in the transfection, it is basically a chemical mediated method. In this, the DNA is mixed. DNA is mixed with calcium phosphate, a substance. It can be any charged substance. So we are taking calcium phosphate over here. This is calcium phosphate or dextran. It can be any charged substance, liposomes, etc. Now this mixture is used as a layer on the surface of cells, on the surface of the bacterial cell obviously. So this mixture, it can be dextrose also, or it can be liposomes. So this mixture is then placed upon the bacterial cell. Now when it is placed upon the bacterial cell, it is used as a layer. It facilitates or it enables the bacteria to take up the DNA. So these are the methods which are being used to incorporate DNA into the bacterial cell. Other method can also be like use of pathogen vectors which are disarmed. Disarmed means now their capacity as a pathogen is made nil. They are first made non-pathogenic and are then used as vectors to allow the infection within the bacterial cell, carrying the recombinant DNA, and then that DNA can be passed on to the bacteria. So there are various methods by which we can incorporate the rDNA within the host cell. So the last method which we will be talking about is the use of disarmed pathogens. And these pathogens are used as vectors, and these vectors infect the cells, and the rDNA is rDNA into bacteria because the vectors carry the rDNA. So this is the method of incorporating, or the bacterial cells are made competent to receive the DNA.